So in class we talked about mitosis, or the process of cell division. We also mentioned that cancer could be the uncontrolled cell division, or it's mitosis gone wrong. So this is what we're going to be talking about tomorrow. So cell division is a very highly organized process which allows organisms to go from a fertilized egg to an adult. And the process of the cell cycle is controlled by proteins. And these proteins are created from the expression of two types of genes. And we have not talked about what a gene is. However, all you need to know right now is that genes are sections of DNA that give instructions. So in this case, these genes are giving instructions for the process of the cell cycle. For the cell cycle, there are two types of genes, and you do not need to know the specifics of them. However, one gene is basically the go gene, and the other is a stop gene. If a gene recognizes that a cell is able to divide and has done things correctly, then the gene will basically say go. If the cell has not done something correctly, then the gene will express the stop code, and it will abort the process and could potentially signal for cell suicide, which we will talk about in a minute, or it will just say we are not going to divide at this point. So in class, I kind of talked about checkpoints. These checkpoints are in the cell cycle along the way. So in interphase, there are a few, and in mitosis, there are actually a few. So there are several systems that the cell can stop the cell cycle if something goes wrong. These checkpoints basically make sure that the cell is doing things correctly before it continues in the phase. Okay, as I said just a minute ago, if there's a stop gene that says no, you cannot divide, if something has gone wrong, this may trigger the cell suicide. The cell might break itself down and cell division may occur from another cell. This cell suicide is actually called apoptosis. Okay, so here are the checkpoints. Remember, G1, S, and G2 are all part of interphase. G1 is while the cell is growing. Before the cell can go into S phase, it has to go past this policeman, this checkpoint. At this checkpoint, this checks to make sure that the cell is large enough and that the environment around the cell is able to support more cells being made. Here it also says that there are growth factors, and these are basically just signals from other cells saying that, hey, we need to keep making more. Okay. Once it passes G1, then it can go into S phase. So remember, S phase is where DNA synthesis occurs. This is where DNA doubles. Once it finishes S phase, it goes into G2. Before mitosis and at the end of this G2 phase, there is another checkpoint. This again makes sure that the cell is big enough to divide and that the DNA has been replicated correctly. If the go gene is expressed, if this little policeman checkpoint says, okay, you've done this correctly, then the cell may go ahead and go through cell division. However, we have this metaphase checkpoint here, which is in the middle of mitosis. Metaphase, remember, is where those chromosomes line up in the middle of the cell. That is where the centrioles have migrated to the opposite ends of the cell and spindle fibers have formed and connected with the chromosomes. This metaphase checkpoint makes sure that all of the chromosomes are attached to those spindle fibers so that in anaphase, each chromosome can be pulled apart correctly. If it passes that checkpoint, then it finishes with mitosis and continues again into the G1 phase. So this is basically normal cells. However, with cancer, cancer is basically mitosis gone wrong. It is a disease of mitosis. These checkpoints have basically been ignored by these cancer cells. They have found a way around it. Okay. This is because of some kind of DNA mutation that occurs in those genes. Now remember, those genes are made of DNA, and DNA gave the cell cycle the instructions. If there is a mutation in those instructions, then the cell cycle may not continue as it's supposed to. So these cancer cells may go ahead and divide as they please instead of following instructions. So some of these mutations can occur spontaneously, but many times it's because of an exposure to a mutagen. Now, humans have actually increased the number of mutagens that we expose ourselves to. An example is UV light. So tanning beds. Tanning beds are not healthy because they use harsh UV light. This UV light can cause a mutation in your skin cells and can cause skin cancer. Smoking is another example. When you inhale smoke into your lungs, you are damaging your lung cells. You're also exposing them to this mutagen factor. And when those lung cells try to regenerate, you're going to have a mutation and it will cause lung cancer. Viruses are another example. Some viruses actually can cause cancer. For example, cervical cancer. You are exposed to a virus. This virus goes in, infects the cells, and then those cells start to divide as if they were cancer. Once these cell cycle genes start to misbehave, it allows the cancer to basically run amok. And a few pictures to show you. This would be bone cancer or osteosarcoma. So a cell in the bone has become, there's a mutation, it starts to divide rapidly. This is an example picture of one. If you look at an x-ray, this is what it looks like, and while that doesn't look like much, it will start to hurt, and it will certainly start to cut into the muscle that is surrounding that leg. Okay, for smoking, for lung cancer, I said earlier that you breathe in the smoke is damaging your lungs. Here in the x-ray, everywhere that you see black, that is basically air. So here is one half of your lungs, and this would be the other half. And here you can see the tumor. This tumor prevents this lung from functioning correctly, which basically inhibits a heavy smoker's breathing, which is why they have very heavy breathing. Alright, this is kind of a really cool situation with cancer, and these cells still exist today. There was a lady about 50 years ago named Henrietta Lacks that had cervical cancer. Some of these cervical cancer cells were removed, isolated, and are actually still being grown in cultures. They have actually helped to make polio vaccines and are still used today in research. So just so you can see the difference between normal cells and cancer cells basically ignore the density-dependent inhibition of growth that are in normal cells. So if you remember back to ecology, we were talking about density-dependent and density-independent growth factors. Cells have them as well. If you put cells into a dish like this picture over here, they will start to divide, but they will only divide until there's no more room for them to divide. So they will create this single layer on this dish. If you remove a few cells, then they will grow back, but then they will stop dividing. Cancer cells do not have that trigger, and so they will just continue to divide. They do not stop when the density has reached a certain point. They do not exhibit this density-dependent inhibition. Right, a lot of you have heard the word tumor, and that is basically this mass of cancer cells. Benign tumors versus malignant tumors. Benign tumors are not necessarily harmless, but they're easier to control because they remain at the original site. Malignant tumors are the ones that are bad. They end up actually sending out signals to your body to create new blood vessels. These blood vessels basically supply this tumor with its own food and oxygen. So these cells can continue to divide without any control. The worst part about it is the fact that when these new blood cells come in and supply the tumor cells can now move into the bloodstream and then move to a different part of the body.
Cancer is a group of over 100 diseases characterized by abnormal, uncontrolled cell growth. In a healthy body, cells grow, die, and are replaced in a very controlled way. Damage or change in the genetic material of cells by environmental or internal factors sometimes results in cells that do not die and continue to multiply until a mass of cancer cells or a tumor develops. Most cancer-related deaths are due to metastasis, malignant cells that penetrate into the circulatory system and establish colonies in other parts of the body. Great advancements have been made, but cancer is still the leading cause of death for people.
Okay, so that basically just showed you this process of a malignant tumor that got started, the cells started to divide. This angiogenesis occurred when those new blood vessels came in and started to supply the tumor. And then metastasis occurred when that cell got loose from the tumor and traveled to the brain in this case. Alright, in order to stop cancer, if you want to stop a benign tumor, usually you can just remove it. Or you can do radiation therapy. Radiation therapy stops any cell that is dividing. So if you have an uncontrolled division of cells and you stop it, that basically stops the cancer. Malignant tumors are a little bit trickier. They have chemotherapy. All chemotherapy is a drug therapy with chemicals. So this is where you see a lot of issues in cancer treatment. Basically, chemotherapy also kills any cells that divide frequently. So any of them. It does not just pick on cancer cells. It will pick on any cell that constantly regenerates itself. So, for example, your intestines, your bones, your hair. That is why people undergoing chemotherapy will lose all of their hair. They are in pain because of the bone marrow. They get very weak, and they have issues with digestion. Okay, once the drug is stopped, these side effects basically disappear, and your hair starts to grow back, and you can start eating normal food. Okay. Basically, chemo drugs stop any cell that is dividing. Okay. If you have any questions, make sure you come ready to ask them in class. Have a good night, guys.